Hi everybody, thanks for joining us today. I am Cyril Laurent, heading Median's North American activities. Median Technologies is a mid-sized oncology-focused imaging CRO incepted in 2002 and providing services worldwide with operations based in US, Europe, and China. We are servicing studies from phase 1 to phase 3, centralizing images, verifying their quality, and organizing central assessments of response to treatment. We are also providing advanced scientific and operational services supported by our cutting-edge technology. We are happy to sponsor this new webinar, part of our regular series dedicated to imaging and oncology. We will be reviewing together different design options for central imaging. To drive us through these options today, we have two experts. The first one is Ileana Gallagher. She has been working for 12 years supporting imaging in clinical trials as project manager. Also with Ileana, we have Diandra Fernandez, who acquired her experience during 1 year of specific data management experience and 5 years of project management. Do not forget you can ask questions during the presentation and they will be reviewed during the Q&A, so you can submit them anytime. Without further ado, I turn the floor to you, Ileana.
Thank you, Cyril. Thank you for that lovely introduction. So hello, everybody, and thank you again for joining us today for this webinar to hear us discuss ways to optimize your clinical trial design. I will provide you with the agenda topics that will be discussed today. We will provide an overview of the study design and how its factors can impact imaging. We will discuss the clinical imaging blinding, as well as the importance of it, and the strict adherence to it that is performed in the central review process. We will detail the central imaging reviews and the different types as well as workflows, the read session organization and how images get presented to the independent readers. We will also detail how quality is ensured surrounding the central reads and the processes as well that we can put in place to promote quality and integrity. Lastly, we will summarize factors on how to choose the right design for your trial. Okay, so our first item here is study design impacting imaging. Imaging certainly depends on numerous factors. So most clinical trials, predominantly within oncology and even CNS, are related to imaging, and therefore can require some independent imaging assessments to be performed. Imaging is an important key factor within the clinical trial. And with that being said, imaging does not come in one size fits all. There are various types of study phases and clinical trial objectives that would ultimately lead to a certain level of imaging required as well as quality. With that, imaging can be the main primary objective. It could be the secondary or even an exploratory endpoint. So that would have a key important role on how you want your imaging to be delivered. Also, there are multiple sites that are involved, and this can produce some potential variation in the images and quality with the different types of consistency. Often many sites are in different locations, and so therefore their capabilities can vary. And so the imaging vendor involved would work closely with the sites to provide study-specific material that would ensure imaging acquisitions and similarity of imaging and modalities remain consistent. Choosing the right modality, whether it's echocardiogram or computerized tomography, is an important consideration. So determining the site's demographics and capabilities is important to the study. Clinical imaging blinding is a very common point when it comes to the independent central reviews. The patient blinding can be a concern often at the sponsor level or with regulatory authorities. So it's used to avoid any bias in the results assessments. The ICRO would typically take every possible step necessary to ensure that the patient confidentiality is kept and that complete blinding always remains the same. A fully blinded centralized imaging interpretation process can greatly enhance the credibility of the imaging assessments and ensure consistency with it. So readers are always to remain blinded to most clinical signals. The FDA does recommend central blinded review in situations where the site image management interpretation can vary. For example, results of the image measurement, which could be important for eligibility, safety, or response endpoints. So this is a key point within the central imaging vendor to ensure that blinding is always consistent and remains the same, and that the readers are never unblinded at any point. All right, and so now the types of central reads, I will hand it over to Deandra to detail the different types of reads that can occur.
Thank you, Ileana. For the next couple slides, we are going to be discussing the frequency of reads, such as ongoing and batch, time-sensitive reads, similar to eligibility of confirmation and confirmation of progression, chronological versus non-chronological reads, the workflow of radiology and oncology reads, independent committee, global review, as well as single reads and multiple reads with adjudication. It is important to note that central reads do not replace site reads and are usually conducted in addition to them. So at first, we're going to be talking about the imaging frequency. The most common form of blinded central read is an ongoing read frequency. This allows the sponsor and the site team to review any real-time response, as well as allows flexibility related to data transfer. So for example, you will receive a time point, and that time point, once it goes through quality control, will be sent over to the reader for them to complete their review. The next type of frequency is batch frequency. This is when you receive multiple time points, and once those time points have been QC'd, then they go over to the reader in order for the reader to complete their assessment. This provides greater clarity and reader focus, as well as a reduction of recall rates. This is a preferred option for retrospective studies. An example of this is if you have 24 time points that you receive, you would do the quality control of those time points and then send all those time points over to the reader in order for them to complete their assessment. The next is partially retrospective. This is when you are moving from a collect and hold study and shifting to a study that needs central review. For this frequency, you could either use a batch assessment or have the assessment be completed on an ongoing basis. One of the reasons that you would move from a collect and hold to a central review study is that your study may have good enrollment, additional funding, an extension of the study, or you had a protocol amendment that now requires central review. One of the cons related to using partially retrospective frequency is that without the ICRO involved at the early stages, the risk can be high as documents may not have been in place in order to collect the images properly for assessment. Okay, now we're going to be talking about prospective versus retrospective. So for a prospective read, it watches for outcomes such as the development of disease during a study period. This study usually involves taking a cohort of subjects and watching them over a period of time. So you would receive the image acquisitions from the site. You will also collect clinical information, and then it would be sent over to the blinded independent readers for their assessment. The next is retrospective reads. This is when images and clinical information have been previously acquired and provided to the ICRO for central reads to be conducted. For these reads, it looks backwards and examines the exposures to suspected risk. So you would collect the image acquisition as well as the clinical information. Reads may have been completed already, and you are going back and performing rereads again as necessary. Okay, now we are going to be talking about time-sensitive reads. So one of the time-sensitive reads is confirmation of progression. You will use confirmation of progression in order to have an informed decision related to the patient's status. Typically, the status would be the patient withdrawing, continuing the treatment within the study, as well as the level of treatment. Typically, this progression is requested by the site or the sponsor. Once it is requested, it is sent to the reader in a timely manner in order for them to confirm whether progression is seen, so the sponsor can create that informed decision on the patient status. The next is eligibility of confirmation. Using this confirmation ensures that the subject meets the inclusion criteria from the central review in order to promote efficacy results. One of the ways that you would use eligibility confirmation is if the population is difficult to enroll. So when you have a difficult population that's difficult to enroll, you could use an eligibility confirmation to ensure that the subjects have met the criteria before they continue on within the study. The eligibility confirmation can be performed as a single or a 2+1 radiology review paradigm. And right now we are going to have a poll discussing time-sensitive reads.
Thank you, Deandra. Okay, so here's a quick poll for everyone in the webinar if you are able to select one of the options here. So the question is, have you experienced a time-sensitive study which included eligibility or confirmation of progression? The choices are often, which could mean 5 or more studies, sometimes, which could be 3 or less studies, rarely, maybe it was only one time for one study, or never, no studies. So we will give you a minute or two to complete your response, and then we will go over the responses. Okay, so it looks like we got some responses. Thank you, everyone who participated. So it looks like the most popular answer is often, which is 5 or more studies, with the only other option chosen after that is never. So what's interesting about that is you either experienced it or you haven't. So I know in my experience, I've probably done 5 or more studies as well. So it's quite interesting that I would align more with the majority here. But if you've never experienced it, it just means you haven't been in this situation before, and you likely will come upon it at some point. But we will also be providing more information on why you would choose eligibility or confirmation of progression for your study.
Great, thank you. And now we are going to talk about the single versus double read model. So for using single or double read, it's really dependent on your endpoints within your protocol. For a single reader, you will have Dr. A, and Dr. A will read the baseline through time point 4 and create a response. So as you can see for time point 1 and 2, Dr. A stated stable disease, for time point 3, partial response, and for time point 4, progressive disease. For a single read, you would use Dr. A's assessment. For a double read, you will have Dr. B, and Dr. B will also go through and read the time points and create their own response as well. So some of the differences here is time point 2. As you can see, Dr. A stated stable disease and Dr. B stated partial response. So this is a discrepancy which can trigger an adjudication. Discrepancies become more likely as the number of endpoints increase. Adjudication is beneficial in order to confirm a reader's assessment and ensure reliability of response. So for an adjudication, you will have Dr. C come in, and Dr. C will look through time point 2 and determine which reader is best endorsed for the response, whether stable disease or partial response. And as you can see, Dr. C agrees with Dr. B, so Dr. B will be the endorsed reader, suggesting that Dr. B's response at time point 2 is the most thorough evaluation.
All right, so now we will go through the imaging presentation to central readers. We will detail the chronological versus non-chronological reads. So for the first one, chronological reads, these are the most common types of reads that are provided to the readers. It's just presenting the images following an agreed time point scheduling that is typically listed in the protocol. This is common and typically a preferred option as it does increase the efficiency as readers are seeing the images as they flow along. So it could mean seeing the progression over time easier by seeing, time point 2 as opposed to time point 3 in some sort of order based on their scheduling. The other type of reads would be non-chronological reads, which can also be detailed as random sequence reading. These would be reads that are presented to the independent readers outside of some chronological order. This could tend to declare PD at a higher rate earlier. So in this way, the readers would get all of the images sometimes all at once, but in not any specified order. So they could be reading, time point 3 and baseline simultaneously, as opposed to reading them in the order that would typically be preferred. This is not common, so it's not often in many protocols or in many study designs to have them this way, but it can be beneficial with certain criteria that may be involved and, dependent on the medical lead's observation of the best way to have images presented to the readers. Another type of imaging presentation to the readers is smart reads. The smart read concept is quite common and probably heard that term before. This consists of reviewing not just the baseline alone, but only when the next available time point is available. So in this way, the main advantage is increasing the quality through selecting the lesions that can remain measurable over time. Sometimes it could be better for the reader to make their assessments seeing one time point and the next one close together. So in this way, they can make a better assessment as opposed to just reading the baseline alone when it's received. You can see the time sequential presentation here in the diagram on the right with a sequential lock using the smart read process. So, and below where it indicates a smart read, you would see the baseline and time point 1, which the reader would read together. The reader would then read the time point 2 and time point 3 follow-up visits as they are received, and time point 4, and the previous time points would then be locked. Without the locking process, it just means that the readers can read the time points as they are available. So the smart read would still be baseline time point 1, but then when time point 2 is available, the previous time points are not locked, so they could always review them as well again. Above, it indicates when there's a final lock, so it would be without reading the smart read process, just reading all of the time points, and then only when the final all images are received, all of them would then be locked. Okay, so on to the oncology and radiology reads. This is a workflow that is quite common when you have both radiology and oncology reads together. So it all starts with the radiology images received. They would be received and a thorough, exhaustive QC would be done. During the QC process, you just confirm that the anatomy is all there, that nothing is missing, and that there's no quality issue, and everything is within the field of view and no part of the anatomy is obstructed in any way. Once the QC is passed, the images would then be sent simultaneously to reader 1 and reader 2. Both readers would need to conduct their separate radiology reviews of the same images. And once they both complete their assessments, a QC is done on both assessments. So it is determined at that point if the readers had the same agreement on the type of disease that they call, or if they had a discordance in their assessments. If a discordance is noted, it would then go on to an adjudicator. The adjudicator would review the images and their assessments and would then confirm whether they endorse reader 1 or reader 2. Once the endorsement is complete, the image would then be completed at that point for the radiology portion. If both radiology readers did agree on their assessments, an adjudicator would not be needed, and it would then be QC'd, and the radiology read would then also be completed. At this point, once the radiology read is completed, the clinical data would then be sent over to the oncologist. So the clinical data is separate from the radiology reads and could be received at any point. It does not necessarily have to be received once the images are available. It could be provided at any point, at times even before the radiology images are received. Clinical data is a separate form that is provided to the oncologist, and the oncologist could perform the assessment once that is available. The data is typically received from the sponsor, already available and QC'd ready for the oncologist. Once the oncologist completes their assessment, it would then be QC'd, and both the radiology and clinical assessments would be completed at this point. It is one thing to note that the radiologists, as mentioned earlier, are both blinded to the patient information. They are not received with any identifying information to the patient, such as location, treatment arm, or any other identifying information, only the images. In addition, the independent radiologists are also blinded to their parallel radiologist identity and the assessment. So at that point, they are unaware that it is being read simultaneously by another reader and or if it went through an adjudication. The oncologists are also blinded to the same information. They are typically only provided with a time point name and or a date. They would have the clinical information accessible to them that is defined in the ICRO documentation. So the oncologists performing the clinical review are also blinded to any of the radiologists performing their radiology review, as well as their identity and everything. Some types of clinical information that can be provided to the oncologists could be prior radiotherapy, chemotherapy, local therapy information, patient status reports, anything that is typically in a data format that's clinical and not imaging related. Okay, so the independent committee, this is goes along with the central review process. So both RESIST and WHO do recommend the use of an independent review committee. This is to ensure consistency and minimizes bias as well within the clinical trials. So it's not surprising that the independent review committee and the investigator site tend to differ at times on the assessments that are provided. There is variability when you are assessing a read response and progression-free survival between both the independent review committee as well as the investigator sites. So if the primary endpoint is on the basis of tumor response, it is recommended to use this type of independency of reads, as it does aim to improve objectivity and reliability of data assessments. It could be that it tends to be subject to observer bias. So the centralized process can better provide a verifiable and uniform independent reader training, as well as ongoing management of reader performance that ensures that data is of high quality and integrity. So in addition to the central readers, that can be the independent review committee. The independent review committee can also be a separate consensus read. It is kind of like, can be a tumor board, which can involve multiple readers or experts that are working together to discuss the image exams or a particular select view. This can be common in smaller trials or potentially for rare or even complex tumors in which the image interpretation can be difficult. So there are two kind of separate kinds of independent committees that we could be referring to. Okay, and on to a global review. So the global review consists of a complete review of all time points and adjustments of measurements. This is in addition to the ongoing reviews that occur within a given subject time point visit. This is recommended prior to adjudication when there is a high number of time points per patient. What the global review does is it just provides a better overview of the overall patient response to treatment. So this can be recommended when there is a registration potential, because a global review can provide an additional layer of quality of the read assessment. This is highly recommended and arguably effective for long-term studies. It can be beneficial, especially when there's a high number of reads, such as more than 4 time points. And during a sequential lock, you can still perform the global review process. So some additional objectives, as I may have already mentioned, is just improves the global patient's assessment through the review of all time points. And this can also identify and isolate any corrections that are needed to the chronological analysis of the patient's response. Because ultimately, when the data is provided, it would be the global review that would have most commonly the overall assessment of each patient. Okay, and so this is kind of, this portrays in images what a global review can consist of and how it would look like. You would see here that reader 1 and reader 2 had both completed their full assessments of each time point for these subject images. And at the very end, once all the time points have been received or prior to an interim analysis or a final data log, a global review is decided to be performed to provide an overall assessment of all the reads. So in this case, you can see from the global assessment that it shows the slow-growing lesions and the date of PD better. This global review offers a better assessment to the date, and it shows it more clearly for slow-growing lesions from time point 2 to the last time point in this example. So it's beneficial to have this, of course, depending on the criteria and the objective of the study, but this, in this case, was proven to be very beneficial to have the global review performed.
Okay, now we are going to be talking about the quality surrounding central reads and ensuring that quality. Some of the ways that you can ensure the quality of the central reads is by utilizing that double read plus adjudication paradigm. This allows to catch discrepancies as well as to catch and correct any reader errors such as missing a new lesion to ensure that the most correct and thorough evaluation is picked for the assessment. Another way to ensure quality is through monitoring. This ensures reliability of the produced data. It also checks the reader performance to address any issues. Another way is to ensure the reader qualification insurance, and this is just meant to ensure that the readers are the most qualified for the specific criteria or indication. You can have the sponsor team approve selected readers and also ensure that the reader is fully trained on the study criteria. So another way to ensure the quality of the central reads is to have inter versus intra variability. So for intra reader variability, this is when a random selection of patients that were already read by a reader are resubmitted to the readers for their review. The new reads are submitted to the readers under a new patient ID in order to prevent bias with the reader thinking that they have already completed these reads. And then you would compare a reader's agreement with him or herself. The other type is inter-reader variability. For this one, there's no need to do additional reads. You would just be comparing a reader's performance against another reader. And you would see their comparison based off of some factors such as the overall adjudication fraction, such as the percentage of subjects requiring adjudication, as well as the endorsement fraction, which is the percent of subjects for which the reader was endorsed by the adjudicator. For these types of variabilities, the potential outcomes are that you can confirm that the reader quality is good, and you can make a decision on whether the reader needs to be retrained, removed, or replaced altogether. Okay, and now we are going to have some points to consider when determining a study design.
Yes, so one point to consider when figuring out what kind of study you want to have and how you want the reads to be performed is, do you want the reads done by a radiologist or only reviewed by a radiologist? In some cases, you can have a technologist perform the reads and then get them centrally reviewed by a radiologist. And this could be an option when a specific type of study is required, maybe a smaller study where you can, it could be a cost-effective option. Having it reviewed by a radiologist is having it both done and reviewed by one as well. So some topics to consider. I know often some studies have done this before, where a technologist will perform part of the reads and then get them reviewed by a radiologist. Could be something to consider for certain trials.
Another consideration is if the trial data is sensitive enough to warrant double reads. As we discussed with the single and double read paradigm, it's really dependent on your endpoints. The more endpoints that you have, the more chance that there may be discrepancy when it comes to the response. So in that case, a double reads plus an adjudication may be the way to go in order to ensure that the best evaluation is being reviewed and used.
Yeah, that's a good point. And also when you're on a phase 3 study, you may choose to warrant double reads and have a global review and all of the bells and whistles, because the data is very sensitive and it might be eventually submitted to the FDA at one point. Another option to consider would be, are there enough patients in case some of them are not eligible for the central read assessment? So oftentimes enrollment is just expected to occur maybe for the first 6 months or possibly even a year. But as patients, maybe the patient enrollment population is not big enough, and at times subjects may become screen failed or ineligible, which could cause enrollment to continue on past 1 year and make it possibly even an extremely slow enrollment for most of the study. So some options to consider is how the criteria that may involve, or the inclusion and exclusion criteria, if maybe that's going to warrant some of the subjects to not be able to enroll, or if you are expecting a small enrollment, some options that you can take to try to eliminate part of those processes. So possibly the patient population pool could grow a little bigger.
Yeah, you could also utilize an eligibility confirmation when the patient population is difficult to obtain, that this will provide efficacy results to make sure that the patient does meet the criteria and is able to continue out throughout the study. So what type of central read is needed according to the phase of the study? So as the studies go from phase 1 to phase 3, the need for review increases, as well as most likely the patient population, the endpoints as well. So when considering this, maybe you could move from a collect and hold to a central read when you are moving toward different phases. You may use the eligibility confirmation or progression of disease confirmation to determine, the patient population if, again, they are difficult to attain. And this is all dependent within your endpoints as well.
Yep, that's a good point. The different phases of the study could cause different types of central reads. I think we mentioned that in one of the points earlier. So that would just make a huge difference from the different phases and what types of reads would be required. And there would be some major discussions as well with your medical team on the types of criteria involved for the specific types of reads that would be needed.
Okay, and here is a study design recommendation table. So this table is just to illustrate some other considerations to consider with your study design, such as interim analysis, limited budget, rescue study. We do have read organization, read paradigm, and other imaging considerations to consider when you do have, like, let's say, a limited budget. If you have a limited budget, then maybe the read organization would be best to use retrospective after collect and hold. For a rescue study, possibly partially retrospective and ongoing with a double read and adjudication for quality purposes.
Okay, great. So now we can look to see if there have been any questions submitted. If you have not submitted any questions, you can still do so. Me and Deandra are here to answer your questions that you may have. So I can go with the first one here that's being sent my way. It says, can you discuss the processes in place at Median to ensure that the data is blinded? Well, that's actually a very good question, how Median handles data blinding to ensure that our readers are completely blinded. So our process is, once the images are received, we do a full QC on them. So before they are sent anywhere, or even provided any feedback to the sites, we do a full QC to make sure all of the anatomy is there. And one of the main things that we QC on is ensuring that the patient, this image is fully de-identified. So if you are sending it through e-transfer, for example, it already helps with that de-identification process. So it would automatically de-identify it. So once we receive it, we know that we don't have any patient information. And as long as everything else within the imaging and the quality is good, it gets sent to the readers. So at Median, we take all precautions to ensure that all of the data we send to the readers are not only de-identified, but that they are of the best quality imaging, and that we ensure that the QC is good so that the readers can make their full assessments. So all readers in any case will never get images with any patient data information, because we would do a full assurance of that it never happens. And as far as I'm aware, it's never happened. So good question.
Yes, great question. Another question we have is what are the time frame for a time-sensitive read? This is something that you can discuss with the sponsor, as sometimes they would want to have it within 24 to 48 hours, but typically it is completed within 3 to 5 days, as this gives you room for both of the readers, if you have a double read and adjudication paradigm, for both of the readers to complete their assessment and adjudication if necessary, if there's discordance.
Yep. Okay, and so another question here is a smart read a standard process? That's another good question. The smart read can certainly be a standard process. It's not necessarily always required, but the sponsor would agree, and so would the medical team, for example, at Median, would agree if a smart read would be the best way to move forward for this specific study. So most often, a smart read would help with confirming the lesion measurements and ensuring the growth over time. And it's also cost effective where, a reader is not just reading the baseline alone, but only reading it with a follow-up visit. So you eliminate one additional read if it's not necessary. So it could be a good option for many reasons, but of course, it depends on the objective of the study and the criteria involved and what type of readers you're going to have and timelines. But additionally, a smart read is a good way to go in my experience, and is certainly a good standard process for many studies as well, especially for RECIST studies.
Yep. Another question I see here is how do you ensure time points get read in order with a batch frequency? So if you recall, batch frequency is when you get a number of time points, and then once you have a set number, let's say 25 time points, and all those time points will be sent to the reader. So in order to ensure that it gets read in a chronological order, one of the things that you can utilize is the Smart Reads feature, as well as sequential lock. So once the reader completes, they won't be able to read time point 2 until they complete time point 1. And that's a feature we have in terms of locking with Smart Reads and the sequential lock as well.
Okay, well, thank you, Ileana and Deandra, for those detailed insights. I think we covered a lot today. So I would like to thank everybody for participating to this webinar. Just as a reminder, you can contact Median Technologies for expert advice on the email that is displayed right now on the screen, info-request@mediantechnologies.com. You can also use the QR code to get an access to our website, mediantechnologies.com, and obviously contact us through social media. We would be more than happy then to help you in providing some expert advice for your clinical trials in oncology. And this will close this webinar. Thank you again to Deandra and Ileana for those deep insights about what are the options for the imaging design in clinical trials. Thank you.
Thank you, everyone.